Formation and composition of bile. Bile is made up of bile salts, bile pigments, and other substances dissolved in alkaline electrolyte solution. Bile salts are synthesized and secreted by hepatocytes. Bile pigments are picked up from blood sinusoids. The secretion of the bile is around 500 ml per day, and it has a yellow color. It tastes bitter. So, the bile is an alkaline juice comprised of water and solids. The organic components mainly bile salts, bile acids, bile pigments, lecithin, fatty acids, mucus, proteins, and the lysosomal enzymes and immunoglobulins, glycoproteins, sometimes bacteria or virus. Then inorganic contents are mainly the sodium, potassium, bicarbonate, and bile is secreted by hepatocytes, so that is why it is called as hepatic bile, and is concentrated in the gallbladder, which is called as gallbladder bile. Bile acids. Primary bile acids are produced by hepatocytes from cholesterol. They include cholic acid and chenodeoxycholic acids. Primary bile acids contain steroid nucleus with cyclopentanoperhydrophenanthrene ring, and they reach the duodenum through bile. In the small intestine and colon, it is converted into secondary bile acids by bacterial action. Secondary bile acids includes deoxycholic acid and lithocholic acid. Bile salts. It is the sodium and potassium salts of conjugated bile acids. They are derived from primary bile acids. It is synthesized in hepatocytes. Bile acids are first conjugated with taurine and glycine, then they form bile salts in combination with sodium or potassium. Functions. They help in the emulsification and digestion of the fat, and thus they are going to help in the absorption of the fat. They stimulate the formation of bile and hepatocyte called cholerectic action, and they stimulate release bile from gallbladder, and it is called as cholagogue action. Laxative function helps in easy bowel movements and stimulate peristalsis. And from route for the removal, it is the route for removal of cholesterol, as they are formed from cholesterol. Bile pigments, these are the excretory products in bile, and they are formed by breakdown of hemoglobin in reticuloendothelial system. Bilirubin is transported to liver along with plasma proteins, and this is called as unconjugated bilirubin, which is with the albumin. This bilirubin is broken down by hepatocytes. Proteins separate and bilirubin conjugated with glucuronic acid to form conjugated bilirubin. In intestine, bacteria act upon bilirubin, and 50% of the bilirubin is converted into bilinogen. Most of the bilinogen enters the liver through enterohepatic circulation, and it is re-excreted through bile. About 5% of the urobilinogen is excreted by kidney through urine. Some unabsorbed part is excreted through feces as stercobilinogen. This gives yellow color to urine and feces. Functions of bile. It is required for digestion and absorption of fat by two mechanisms: by activating pancreatic lipase, and it is going to help in emulsification of fat and formation of micelle, and it is going to help in the absorption of fat. So it is also helps in absorption of fat soluble vitamins. It has a cholerectic and cholagogue actions, and it is endogenous synthesis of bile salts. It's important for synthesis of bile salts. It has a bacteriostatic action, and it is a major route for loss of cholesterol from the body, and it has a lubrication function due to the presence of mucus and alkaline nature of its helps in neutralizing acidic chyme. Coming to enterohepatic circulation, about 94% of the bile salts are reabsorbed into blood from small intestine. That is, about one half of this is by diffusing through the mucosa in the early portions of the small intestine, and the remainder by an active transport process through the intestinal mucosa in the distal ileum. They enter the portal blood and pass back to the liver. On reaching the liver, these salts are absorbed almost entirely back into the hepatic cells and are resecreted into the bile. In this way, about 94% of all bile salts are recirculated into the bile, so that on the average these salts make the entire circuit some 17 times before being carried out in the feces. The small quantities of bile salts lost into the feces are replaced by new amounts formed continually by the liver cells. This recirculation of the bile salts is called as enterohepatic circulation of bile. This circulation is necessary because limited pool of bile acids. So this diagram showing the enterohepatic circulation, the the bile salts which are produced in the liver, they are secreted into the intestine, and in the distal part and the colon, they are almost 95% of these bile salts, they are reabsorbed into the portal system, and they are taken up by the liver and uh, resecreted back into the GI tract, and this is called as enterohepatic circulation.